Vacuoles. This is fluid-filled sacs for storage. A big, big central vacuole like that you only find in a plant cell, or maybe in a protist cell, I guess. But you certainly never find them in animal cells. They are small or absent in animal cells. There are no vacuoles in bacterial cells because, of course, they're membrane-bound, and you don't have any membrane-bound organelles in bacterial cells. Plants have a large central vacuole. And so this is a central vacuole, and its main job is to provide or to help maintain turgor pressure. Never I know how to spell that thing. I think it's an ER, turgor pressure. Anyway, they help maintain turgor pressure, which means it keeps the cell crispy, if you want, like a celery that's not droopy. They help to maintain the water pressure in here, and so we're going to spend a lot of time on that with our next unit. Plant vacuoles. They include storage of sugars, amino acids, minerals, lipids, waste, salts, water, and enzymes. So they can store tons and tons of stuff, but the water is a pretty important one. During growth, fluid pressure builds up in the vacuole, forcing the cell wall to enlarge, so they help the cell to grow the right way. The central vacuole is important for turgor pressure. I guess it's with an OR. So here's a turgid cell, which means that water comes in and it gets really a lot of high pressure in here because there's so much water in here and it pushes out here. So that's when you have a nice crispy piece of lettuce. If you have water leaving, look at how your central vacuole in this plant is so tiny. This is a floppy or a flaccid cell. So water's lost and the vacuole shrinks and the cell loses shape. This is when you go outside in the summer and all the plants look droopy and your mom says, "Oh my gosh, can you water the plants?" You want to change them from here to where they should be like this. So you want a nice full central vacuole. Contractile vacuole. To contract means to make something smaller and shorter. So a contractile vacuole is found in unicellular protists like this paramecium, just for an example. So here's the contractile vacuole, and I'll have to show you the movie on the next page in school because I don't think that'll come out. But anyway, this regulates water intake by pumping out excess water. So it maintains homeostasis in that way. This will also make sense more in our next unit, so I'll talk about it there more, but I do need to just cover all the organelles briefly. So if too much water comes in here, there's no cell wall, this thing will burst. And so the contractile vacuole forces water out. So this keeps the cell from lysing, which means bursting. To lyse means to burst. And so if I don't know if this is coming out, it probably isn't, but this is a contractile vacuole animation and the water is being squirted out of here. So I think the time you've heard this before is, at least for my honors group, I used the word hydrolysis. And that was using water to break something. This is lysing, which means the cell will actually burst. So water's going in, and if you don't push water out, it'll break. And so that's what this is for. So that would not be a problem in plant cells because plant cells have a cell wall to keep them from breaking. Quiz time. The endomembrane system includes the rough ER and the smooth ER. Oh, sorry, which is false. I didn't even see that part. So the endomembrane system includes the rough and smooth ER. It does do that, so that's not false. So I would say that's true. The endomembrane system divides the cell into compartments. That's true. You have the compartment where what's inside the ER and a compartment with what's inside the Golgi and a different compartment for what's inside the lysosome. So that's true. In fact, your cell compartments are super, super important. You don't want the enzymes in the lysosome to be spread throughout the cell or they'll actually kill the cell. So these compartments are a super, super big deal. It's sort of like at home you have a jar for your marjoram and a different jar, like for all your spices, a different jar for your cumin maybe, and a different jar for, or cumin, is that it? And a different jar for your basil. All the different jars have to keep the stuff that's in them separate, and you wouldn't want to mix that with your jar for baking soda, for example. Okay, so anyway, I hope some of you cook. The endomembrane system is a system of interrelated membranes that are all physically connected. That is not true. You can have a Golgi body that is not connected to a lysosome, although it was at one point, but it's not anymore. So this should be the answer. The endomembrane system is involved in the synthesis, storage, and export of important molecules. That's true. So your answer here is C. Based on their function, you would expect melanocytes. Okay, never told you about this, but don't worry because I am right now. And the MCAS loves to do that. The MCAS loves to throw stuff at you, and what you have to do is not panic. So let's not panic. Okay, new word never heard before. I'm panicking. Okay, melanocyte cells. Okay, so a melanocyte is a cell that produces the protein melanin. The important thing here is that it's a protein. So based on their function, you would expect melanocytes, cells that produce a protein melanin, in the skin to have a higher than usual number of. Which of these things do you need for protein synthesis? So that's what this question is asking. The MCAS loves to, instead of say what's important from protein synthesis and giving you this, the MCAS loves to word it this way. So what's the function of melanocytes? And you're going to panic and say, I don't know what a melanocyte is. They tell you everything, and protein's the key word here. Which of these things are involved in protein synthesis? Not chloroplasts, they make sugar. Not microtubules. Microtubules are proteins, but they don't make proteins. Not lysosomes, they break stuff down. So ribosomes, those are involved in making proteins. So do you see that you knew this answer, but it was worded really hard? Kind of mean. Contractile vacuoles. Make sure you have this in your mind as a separate thing from a central vacuole. A central vacuole is a thing inside a plant cell that maintains turgor pressure. So there's a central vacuole. And remember, a plant cell has this big, thick cell wall. So water comes in here, and that's a good thing. You want it to be turgid. Contractile vacuoles, this is in something that doesn't have a cell wall, like a protist. Water comes in, and if water keeps coming in, this thing's going to break. At some point, the cell membrane just breaks. So contractile vacuole forces the extra water out. So contractile vacuoles stimulate the absorption of salt water. Nope. Help in the excretion of excess salt. Nope. Allow organisms to avoid dehydration. Nah, you're trying not to burst. Are generally found in protists that inhabit salt water. Oh, I really need to do the next unit for you. That's not true. Prevent cells from bursting. That's it, as a result of the influx of excess water. First of all, let's make sure you understand the vocabulary. Prevent, that means stop cells from bursting or breaking as a result of the influx. That means stuff coming in of excess water. Too much water is coming in. A contractile vacuole will send that water out. Goodbye, water. So that's the one. This whole thing of protists in salt water or protists in fresh water, that's super important, and we do that in our next unit, so I apologize. I probably shouldn't have thrown that at you in this one. Okay, the oil from the sebaceous glands. Okay, now is time to panic. I'm on an MCAS, I'm reading a question, and I have no idea what sebaceous glands are. Let's keep reading anyway. Oil, oil, oil. I do know that oil is a lipid. I wonder if that will come in handy. The oil from the sebaceous glands in the skin is produced by which of the following cell organelles? Well, I know the organelle that has to do with lipids is the smooth ER. Oh, look, and that's one of the answers. Hip, hip, hooray. Secretory proteins. Okay, I haven't told you that yet. To secrete something is to send it out of the cell. So here's a cell. If you're secreting a protein, you're sending it out of the cell. So that's a secretory or protein. Secretory proteins are chemically modified in the nucleus. No, 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 no. Proteins don't go into the nucleus. Nope, 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 nope. Unless it's part of a ribosome being assembled or something, or a histone that is involved with the chromosomes. You don't, in general, send proteins into the nucleus. Secretory proteins are incorporated into the mitochondrial membrane. Nope, they're secreted out of the cell. And again, if you didn't know the word secreted yet, don't worry about it. You're learning it right now. Are sent out of the cell? Yep, that sounds like the right one. They're produced by ribosomes on the smooth ER. There's no such thing as ribosomes on the smooth ER. It's smooth because there's no ribosomes. Are produced by the cell for internal use. Nope. So that wasn't really fair. I haven't told you secretory. But anyway, you might want to write that down. To secrete means to send out. I hope I added that to my vocab list for you. Which of the following statements about lysosomes is false? Lysosomes are those teeny little organelles that lyse stuff. They break bacteria. Lysosomes destroy, oh, I kind of gave that one away. Lysosomes destroy harmful bacteria engulfed by white blood cells. That's actually, ooh, what's wrong with this? That's wrong. Um, that goes up here. So the right answer is actually A. Sorry, folks, I have to move that. There we go. Lysosomes synthesize proteins. No, it's ribosomes. A lot of times students confer, and apparently I messed that up. Blah. Ribosomes. See how they both end with ozomes? Ribosomes synthesize proteins, yes. Lysosomes recycle material within a cell. Oh, that's true. Oh, look at that. I messed up. The answer is we're looking for what's false. Lysosomes destroy harmful bacteria. That's true. Lysosomes recycle material. That's true. Lysosomes help digest worn out or damaged organelles. That's true. Lysosomes fuse with food vacuoles, like in a protist or something, to expose nutrients to lysosomal enzymes. That's true. This is the one that's false. The answer is B. I do have the smiley face in the right place. So it's not lysosomes, it's ribosomes that make or synthesize proteins. So this is the false one. Oh man, those false questions are rough. I'm even messing them up. When a cell is deprived of oxygen, so it doesn't get enough oxygen, so that means it can't do cellular respiration. Its lysosomes tend to burst and release their contents into the cell. Lysosomes have digestive enzymes. That can't not be a good thing for the cell. As a result of this, the cell will undergo self-digestion and die. Yeah, that's kind of a bummer. Undergo cell division? No, that happens when things are going well. Produce replacement lysosomes? Well, if only one of them broke, maybe. Produce additional ER? What does that have to do with anything? Recycle damaged organelles? Well, they would, but not if they're going to burst. So the answer here is undergo self-digestion and die. The only time that's good, it is good sometimes. So this is a tadpole. And you know how tadpoles grow, right? They become froglets, so they get little arms here, and they'll eventually start to make little legs, and that tail will go away, and here's the arm, and oh my gosh, it's kind of pathetic. And eventually, anyway, you're going to get this thing called a frog, ribbit. Oh, that is like the ugliest frog I've ever made. Anyway, the way that you get rid of that tail, it doesn't actually break off and float to the bottom of your pond. It actually gets reabsorbed. See how it got smaller and here it's gone? So you had lysosomes break in these cells and the nutrients got reabsorbed inside. Cool, huh? It's true. It is totally cool. Okay, last one. A manufacturing company dumps its waste into a nearby pond. Oh, that's not good. One of the wastes is found to paralyze contractile vacuoles of a certain protist. Okay, so here's your protist. Remember, the contractile vacuole is supposed to force water out. A biologist looking at individual samples of these organisms taken from the pond would find that they are surviving but unable to reproduce. So let's see, if you have a bunch of water coming in and nothing to force it out, do you think you'd survive? No. They've lost water and shrunk? No, that's not the problem. They've gained water and burst. That's it. You have so much water coming in and none can make its way out, so you burst. Bummer. They died of malnutrition? No, they died because they burst. Have died because wastes have built up? Nope, they died because they burst. Hate that. Okay, so that's it for now.